So the previous bit of information were techniques on how to separate proteins in a mixture, either via chromatography or via gel electrophoresis, or the crudest form, which would just be the protein precipitation with the ammonium sulfate salt. Okay, so once you maybe isolate the protein that you were looking for, because remember back, there was an image I had of a cell and I said, I just want all these little green 4-leaf clover looking proteins out of this whole mixture of stuff. Maybe then you want to know what's the sequence of amino acids in of that protein that we've purified. So these are some techniques that are used commonly to determine what the sequence is of proteins. So the very first one way you can do is determine the sequence by shooting it on a mass spectrometer. Okay, and I'll show some examples of that. You also can perform chemical or enzymatic digests, and that's a different way. We'll look at that, some examples of that. And then the third most common sequencing technique for amino acid sequence is you would go back and look at the DNA sequence, right? Because we know that every 3 DNA molecules that convert to an RNA molecule make up a codon, and then on your tRNA, you've got the anticodon, but you know, this codon corresponds to an amino acid. Okay, that was the Nobel Prize awarded for the discovery or figuring out what that patterning was, what the 3 DNA, what the codon was that paired up with each amino acid. So first we'll start with the mass spec technique. So mass spec is incredibly accurate. In fact, the little sheet that Dr. Fraley had prepared for you guys with just some information about the different amino acids, like their mass and whatnot, and some tidbits about them. Mass spec has almost infinite significant figures. Like, it's just a very, very accurate technique. So the question then is, given there's 20 common amino acids, let's say that only these 20 show up in the protein we're analyzing. If you knew the total mass of the protein, would you be able to deduce which amino acids and how many of each make up that protein? So on average, we say that the average amino acid weighs 110, but this question really starts making you wonder, but do all 20 amino acids have a distinctly different mass, or do any of them have the same exact mass? So, for example, like serine and cysteine. Do those have the same mass? No. In fact, no, they don't. They just have a completely different chemical formula that makes them up. Like, one's got a thiol, one's got the alcohol. So yeah, those are different masses. So you would almost think that, yes, if you knew the total mass of a protein, you should be able to mathematically figure out a way to say how many of each different amino acid would be found in that protein, because there's only a certain number of ways those numbers could add up with practically in infinite significant figures, you know, like just think 6 decimal places worth of accuracy to figure out what the composition is. But you'd be wrong because of leucine and isoleucine. So leucine and isoleucine have the exact same chemical formula, therefore they have the exact same mass because they're isomers of one another. So you could, if you know the protein's total mass, you'd be able to figure out the majority of the composition, but you wouldn't be able to differentiate how many leucine versus isoleucine are in that protein. You would just know that the leucine and isoleucine content is this amount of amino acids. Okay, so that's like maybe the one tiny caveat of using mass spec for determining the sequence, is the leucine isoleucine issue. Okay, so probably the most typical way that protein samples are sequenced is through what's called MALDI. This stands for Matrix Assisted Laser Desorption Ionization. You essentially take your protein mixture and mix it with a matrix, dry it on this little bed, and then a laser is pointed at that sample, which essentially ionizes some of the sample, which then travels through. This is a time of flight detector, so smaller pieces are going to travel farther, larger pieces are slower, and then you get this detector here, and the detector would be coupled to a mass spectrometer. So it would tell you the masses of these fragments that are hitting the detector. Okay, the laser, if it has high enough energy, should technically break some of those peptide bonds that are holding together our amino acids in that one protein strand. So what you find is when you look at the mass spec data, so mass spec data tends to look like this. This y-axis is usually relative abundance. And then, yeah, that's what it is. And then the x-axis, though, this axis is... sorry, the x-axis, oh, I'm trying to find my spotlight. So the x-axis here is mass. So you find where your peaks are, and those peaks have a corresponding mass. And what you can do is you find kind of the taller peaks, and you can subtract out the mass from these two and essentially figure out what amino acid corresponds to that exact mass. Okay, so this data right here, this 0.5, it doesn't look like there's infinite significant figures, but I promise you, if you were looking at the screen, you could like click on this mass and you would see all the decimal places behind. Okay, so if you were to take, though, these two big tall peaks right here and subtract 699.5 from this 829.5, and if you had a list of the amino acids, which you kind of do because Dr. Fraley has provided it for you, you could find that exact mass, and you would find out that that corresponds to asparagine, N. So that is N. And then you find the next biggest peak, and you would subtract these two peaks, and you would find out that that mass, that's the difference between those two, corresponds to the glutamic acid. So now you know the sequence already, something that's going N and then E, and so on and so forth. So you subtract the masses from one another, and then you compare it to the known masses of the different amino acids, and you can deduce what the sequence is of your polypeptide chain that way. So that's figuring out what the sequence is of your protein using mass spectrometry. Let's look at a different way, which would be the enzymatic method. So when we perform the enzymatic method, there's a lot of different chemicals that we're going to have to that play a part here. So the one chemical is either BME, which stands for beta mercaptoethanol, or DTT, which is dithiophthalate. Both of these are reducing agents, and they reduce cysteine bonds. So remember, when we talked about the different amino acids, one interesting fact about cysteine, the free thiol, SH, is that in three-dimensional space, if two cysteines are close enough, they can form a covalent bond, which is called a cysteine bond, or sometimes it's also referred to as a disulfide bridge. Unfortunately, in biochemistry, there's most of the time, there's two ways to say everything. So you can have a cysteine bond or a disulfide bridge. Both are correct. Okay, but so beta mercaptoethanol or DTT will reduce that. They'll cut that covalent bond and give you your free cysteine residues back. Okay, so you might have to do that first. You could perform what's called an Edman degradation, and this technique cleaves the N-terminal residue. Okay, carboxypeptidase is an enzyme that cleaves the C-terminal residue off of the peptide chain. Cyanogen bromide cleaves after methionines. Trypsin is an enzyme that cleaves after positively charged side chains, K and R, so lysine and arginine. And chymotrypsin cleaves after big, bulky, UV active compounds, W, Y, and F. So tryptophan, tyrosine, and phenylalanine. So let's look at these a little more specifically here. Okay. I don't think I have mentioned it specifically yet, so I will right now in time, and we'll see this a lot in the beginning of chapter 4. But when we talk about this peptide sequence, it always is going to go N-terminal to C-terminal, always. It's never going to be the C terminal to the N terminal. It always starts with the N terminus and ends in the C terminus, okay? So just keep that in mind for some of these other enzymes that are coming up. But I just feel better now that I've gotten that off my chest and I've said it to you. And I lost my cursor again. Close. There we go. Okay. So the first ones to look at is chemical digest. So if you want to reduce these cysteine bonds, so see, here's one cysteine residue, here's the second cysteine residue, but they have been oxidized together and they have formed this disulfide bridge or this cysteine bond. You can add, this is the beta mercaptoethanol. So that's ethanol. And that would be the alpha carbon. That's the beta carbon. Hence it's beta mercapto referring to this thiol. So beta mercaptoethanol. And that's just going to reduce and break apart that disulfide bridge. And it's going to link up two of these beta mercaptoethanol molecules together. It's kind of just like a do-si-do of covalent bonds here. Okay. The other compound is DTT, or that dithiothiotol, and that's what this compound looks like. But either of those two, they're going to go into your protein and they're going to cut all of the disulfide bridges in your protein. Okay, so the Edman degradation, remember this tells you what the N-terminal residue is. Sorry, the N-terminal residue. And you can use the Edman degradation for about the first 30 amino acids in a polypeptide chain. Remember, the average protein is about 300 amino acids. So for about the first 30, or the first 10% of the total number of amino acids, you can use Edman degradation to tell you what those amino acids are. And so kind of what you would do there is you add your enzyme with your isolated protein that you want. You give it a little bit of time to incubate, and then you're going to run it on an LCMS, which stands for liquid chromatography. Actually, just LC, not MS. Just a liquid chromatography, sometimes HPLC, high pressure liquid chromatography or high performance liquid chromatography. And it essentially just separates out the amino acids on a column, it's chromatography, over time. So you can run your standards. So here are all of the amino acid standards, and then you can run your reaction, right? So if you incubate the this PITC compound with your protein, you'll see whoop, only one peak should appear, and that peak should end up corresponding to an amino acid that's in your standards. So you could say, all right, so the very first amino acid is methionine. You take your protein again, and you add another equal molar amount of this PITC, and you run it on the LC, and maybe the next peak comes and hits right here, boom. And so you know, oh, my second amino acid is histidine. And you can rinse and repeat. So this works for up to about the first 30 residues or so, because you'll start to see some background peaks occurring, because not every single one polypep protein in your solution maybe is knocking off the N-terminal residue with the time you're giving it. So it gets a little murky. But like I said, so the first 30 amino acids you can use for this. Carboxypeptidase is an enzyme you can incubate with your protein, and also it'll just cleave off the C-terminal residue. So Edman degradation tells you what's the N-terminal sequence coming over this way, and carboxypeptidase cleaves off and tells you what's kind of on the C-terminus of your polypeptide chain. Same idea, where you just incubate the two together, and then you run it on an LC, and you can say, oh, look, boom, the last amino acid on the C-terminus is alanine. This one usually only works for one and done. It's not like you can repeat it, just because it gets too messy to try to separate carboxypeptidase away from your protein of interest. That gets too hard to do. So this just tells you the only one time the C-terminal residue on your polypeptide chain. Another chemical treatment, cyanogen bromide, will cleave after methionines. Okay, it ends up, so if we have kind of a slew, here's our little amino acid fragment right here. Here's the N-terminus, then it goes G, R, F, A, K, M, W, V, C-terminal residue, right? Cyanogen bromide is going to cleave after methionine, not before methionine, after. Another way to say it cleaves after is it cleaves on the C-terminal side of methionine. So just how the C terminus is on the right side of the chain, this would be the C terminal side of methionine, and this would be the N terminal side of methionine. Okay, so you could say also it cleaves the C terminal side of methionine. Okay, but once you add your cyanogen bromide, it ends up producing, it reacts with that side chain, which is a like a it's not a how would you call that? It's like a sulfur, but that's an ester, right? It's got carbons on either side with the sulfur in the center. But you end up forming this peptidyl, this homoserine lactone, and then a new N-terminus. Okay. So that's kind of the reaction there. But all you need to know is that cyanogen bromide cleaves after methionines, and it'll tell you something about your new N-terminus. And this makes a lot more sense when we have a practice problem in front of us. Okay, moving on with more peptide enzymatic digest. So trypsin is another enzyme, and trypsin is going to cut or cleave the C terminal side of positively charged peptides like arginine and lysine. So it's going to cleave after anytime there's an arginine or a lysine in your peptide chain. Trypsin will cleave that. Sometimes, kind of like what you'll see in sample problems, it'll say trypsin digest yields 3 fragments. So if a trypsin digest yields 1, 2, 3 fragments, you would know that there must be 2 positively charged amino acids, either 2 lysines, 2 arginines, or 1 lysine, 1 arginine, but there's a total of 2. So that's kind of how you're going to piece backwards and figure out what the sequence is from like these kind of cuts. Okay. Chymotrypsin is kind of a cousin of trypsin, but it just has a different substrate that it recognizes. It recognizes F, Y, and W. So it recognizes those big, bulky, UV active side chains. Okay, so it cleaves again on the C terminal side of that. So anytime you see the phenylalanine, a trypsin, or a tyrosine, you would get chymotrypsin to cleave after that. Okay. When you perform multiple digests and are given a little more information, like trypsin digest yields 3 fragments, and the 3rd fragment starts has this sequence, trip-glue-vail, something like that. Then you end up piecing together what the different sequences within some of these fragments, and then you can comparatively line things up and figure out what the overall sequence is of your polypeptide chain. Okay, again, that makes way more sense in a practice problem, and don't worry, I have one for you to work. So I'll assign that as a quick homework over the weekend to kind of look at for chapter 3 assignment. And they're fun, they're kind of fun. They're like logic puzzles. So, and you can usually Google online people working them out. I'm sure Khan Academy has somebody working one out for you. But I say give it a whirl. If you know what you know about these things. So trypsin cleaves after positively charged. Chymotrypsin cleaves after big, bulky residues, UV active residues. Cyanogen bromide cleaves after methionines. And carboxypeptidase tells you the C terminus. Edmond degradation tells you what the N terminal residues are. And don't forget the reducing agents that can reduce the disulfide bridges or the cysteine bonds. Okay, so maybe you find the sequence out and you figure it all out. So what can you do with these protein sequences? Well, you can perform these sequence alignments, okay? So, and you can reveal via these sequence alignments some evolutionary cousins. You also can sometimes find catalytic important residues. Okay, so if you compare, so this is just a slew of, it's a region of one protein, and we're looking at it across a variety of animals. Humans, chimps, you would probably expect those to be fairly similar, but humans to cow, humans to dog, humans to whale, right? Humans to zebra, rabbit, horse, so on and so forth. You might expect some divergence between this protein here. So how do you do this? You would compare, you scan, and you look for things that are conserved, meaning they're the same, and things that are non-conserved. So this G is actually this G, D, V, E, K, G, K, K, I, F. Okay, so these first 10 residues are conserved. Oh, wait, except for right here, that this one's a phenylalanine instead of the E there. But almost everything else is fully conserved throughout. So if you find things that are conserved against all different types of of animal species, that usually means that that's an important part of that protein. It either has some type of structural function or catalytic function. Okay, maybe not so much this E because it's mutated to a phenylalanine. You can also talk about the types of mutations you could see against these sequence alignments. So you can have what's called a conserved mutation, and that would be a mutation of an amino acid that's still similar. So it's a conserved mutation. A great example of that would be like a D to an E, right? Those are practically the same amino acid side chains. They just differ in one little tiny methylene group. Serine to threonine, again, is a conserved mutation. Any of the hydrophobics, so valine to leucine or valine to isoleucine or alanine to valine, those are conserved because it's still maintaining what the functionality is of that side chain. So those are considered conserved. You could also just have I don't think it's called non-conserved mutation, but just like a mutation where it's just something totally different. Oh yeah, there it is. So conserved mutations are likely important. Regular, what we call mutations, are areas that tend to be not important. So if there's like mutations along these amino acids, that probably means that that spot in the protein is not really important because it has so much variety in it. Okay. And again, like I said, a conserved mutation can still show likely an important region because you're maintaining the functionality of that side chain still. So, the negative charge might still be there, but it's not 100% always the case. So sometimes on some previous exams, I might give a sequence spot like this, and I might say, identify a conserved mutation. So you're scanning and you're looking for a conserved mutation. Again, it's like serine to threonine is a great one. D to E is a great conserved mutation. Two things that are similar is the amide ones, the glutamine to asparagine. So N to Q would be another really great conserved mutation. If you find the hydrophobes, that's fine too. Ks to Rs, we'll count it because here we go. Here's Ks and there's an Rs because, again, we're holding that positive charge there. Like Fs to Ys are okay. Those are practically the same thing. You just add the extra OH group on. Oh, here's an example of this is just a straight mutation. Look at this very last amino acid, a lot of glycines, boom, lysine. So we're going from the most flexible side chain to lysine, a positively charged one. Oh, that's a mutation right there. So this means likely this spot of the protein probably plays not a significant role in some way. All right. What you can do by comparing those sequences is you can end up seeing evolutionary divergence just from the amino acid sequences themselves. You build these trees mostly using DNA sequence of genes, but you can also do it with proteins. It works the same way, right? So things that tend to have areas, regions that are more conserved are going to be on one branch of the phylogenetic tree, and things that are just kind of really different are going to be on another branch of the phylogenetic tree. So maybe even like on this example back here, just looking at this last row where most of them are G and 2 of them are K, I would throw horse and zebra off on their own branch, and I'd keep all these other ones so far on a similar branch. Okay, so that's kind of how that works. And yeah, the levels can get really complex, right? But that's essentially kind of what I want you to think of if I asked you to build a phylogenetic tree from small amino acid sequences. So you're looking for conservations amongst the sequences, and if things are just really off, they're on their own separate arm. I think, and so if things are like semi-conserved, they're on a similar arm. Okay, well, that sums up how you determine what the protein sequence is and kind of the significance of why would you even care what the protein sequence is. So what can you do with that information? You can identify important residues. by performing those sequence alignments, and you can end up building evolutionary phylogenetic trees.